So first of all, everything that I'm going to, all the software, everything that I'm going to show you is completely public domain. So there are no restrictions, no license required, to use any of this. The Netscape program, there is a registration. First time you open it and try to run the program, it will ask you to register. It's not mandatory. You can skip the registration, but we do ask you to register. We don't, harass people with emails. We just want to sort of, keep track of how many people are using the program, and it helps us when we, basically ask NIH for money. So if you can, please register. Okay, so I would like to start here, and this is a Netscape web page. I showed you a slide earlier, and this is a live page. And so a couple of things that I would like to point out here. So there are several links here that will help you. So first of all, I mentioned that Netscape is a plugin to Cytoscape, and I would like to spend a couple of minutes now talking about Cytoscape. So, like I said before, Cytoscape is an open source general purpose network analysis and visualization tool. Okay, and it has been developed and is being used to visualize very different networks, not necessarily biological networks. Okay. So it can be a social network, and it can be anything else, but it is incredibly powerful. And so it's a framework. And so there is sort of, core Cytoscape software, if you will. And the way it was designed is proved to be very useful because, and it is very open. And so it is sufficiently easy to customize Cytoscape to basically write custom software using that framework. And this custom software in Cytoscape world, they're called plugins, or now I think they're becoming more modern, so they're called apps. So you might say that Netscape is an app for Cytoscape, okay? And so there is a very large community of developers who develop various plugins for Cytoscape, and it's an open source community, and people interact, and they have Cytoscape conferences every now and then, and so on. And so it has pluses and minuses, that it is very easy to develop custom software or your own plugin. So the plus, obviously, is that people are using it, and more. And if you're generally, familiar with Cytoscape, because I have to warn you, there is a learning curve there. But if you're familiar with it, then you can easily download another plugin, just install on your computer, and get going in no time at all. That's a plus. Okay. Well, the downside is that there's nothing to stop someone from developing their own plugin, which may be doing the same thing as the plugin that I'm working with. And they sort of do similar things, but they don't know about each other. They don't talk to each other. And that, can be sort of a downside. Okay. So in an attempt to kind of make these plugins better talk to each other, the core Cytoscape, they're undertaking sort of major revamp of software. And so in going from Cytoscape version 2 to going Cytoscape version 3, they have changed a lot of things. Now, this is a good time for me to say that Netscape version 2, which is what we will be using here, is using Cytoscape 2.8, and it will not work with Cytoscape 3, because I just had a conversation with somebody a few days ago, and so it does not work with Cytoscape 3 just yet. So for us, basically, it was not practical to convert this older version of Netscape to Cytoscape 3, because it would be a significant effort. And we do realize that we want to add a lot of features, and I alluded to some of them earlier. And so we figured that, it would be a good time to do it in Netscape version 3. Okay? And again, just to reiterate what I said earlier, we are always open and looking for feedback. And I sort of, assume that if you guys are here, you're interested in metabolomics, your various aspects of sort of, getting experimental data, analyzing the results, and so on and so forth. So you are exactly the people who provided, useful, can provide us useful feedback. So you're exactly the kind of audience we're looking for. Okay. So anyway. What else can you sort of, find on that Netscape webpage? So first of all, there is a link here that says download the latest version of Netscape. Well, before you go there, actually, so if you go here and click on the Cytoscape link, which I'll do very quickly, it will take you to cytoscape.org, where you can get, you can download Cytoscape. Okay, and once you have done that, I'll show you in a minute when I open Cytoscape how to install the plugin. I mean, the Netscape plugin. There are multiple ways of doing it. But what I want to mention right now, if you do not, for whatever reason, if you do not have administrative rights for the computers that you're working on, and you don't want to install Cytoscape there, you don't have to. What you can do is you can use this link instead. That will basically launch Cytoscape with what is called Java Web Start. So Cytoscape will not be permanently installed in your computer, but you still can run it. Okay? So these are different options. Okay, the other things that I want to point out here, and I will go through this in some detail, there are sample input files so that you can try, and you can see what they look like, and I'll show that in a minute. Also, I want to sort of draw your attention for a minute to this other tab, and there is a rather detailed Netscape manual, and you can look at that. It's extremely difficult to keep it up to date, so I think it's sort of, mostly up to date on the functionality that we have in the tool, but there may have been some smaller changes since we did that. And so this is a link to the video tutorial that I mentioned. Actually, we were able to, restore it. It wasn't there this morning. And so this address, we get that email, I get that email, and all the developers get this email. So if you have any problem or any issue, any question, feel free to email. Well, you can email me directly, or you can, if it's Netscape related question, it's better to use this address, because then, somebody will be able to, help identify the problem. And again, there are examples of the input files. Okay, so that's what I wanted to show you about the web page. Okay, and then, with this, I have Cytoscape installed on my computer, and I would like to go to and just, open Cytoscape. So the version that I have is 2.8.2. I believe there is 2.8.3, which Netscape will also work with. So as long as it's version 2 of Cytoscape, it's okay. You don't want version 3 for sure. Okay. So, and I said a few things a little bit earlier about the sort of overall layout. Okay, so this is what the blank Cytoscape sort of, window looks like. So your networks that you're going to build are going to be right here. And the list of networks is going to be here in that window. This is the navigation pane. It will be easier for me to show that once we get there. And so this is what is called data panel. And so when Chuck was showing, specific sort of, data, experimental data on some of the compounds earlier, that will show up right here in this window. So, okay, so so once you have installed Cytoscape, you will on your computer and open it for the first time. You then have to go to Plugins menu, okay? And so as you can see, I have a whole bunch of different plugins installed. Here, you will not have those, but what you will have to do instead, like I said, there are multiple ways of doing it, okay? So one is you can download what is called a jar file from Netscape, from Cytoscape window, but the easiest way to do it would be go to Manage Plugins, and so you will notice that it is trying to connect to Cytoscape website here, and it connected successfully. Just to point out, in order for all of this to work, you have to be connected to the internet, right? Don't forget. Very important. Okay, and so what you can do here, you can search for Netscape and click search, or just hit enter. And so it is finding the plugins, but I mean, it says currently installed because I do have it installed. Okay, so if you don't have it installed, it will just give you an option, and you will click on it, and it will install the plugin. Okay, so for now, I'm just going to get out of this. Any questions so far? Okay, let's move on then. Okay. So then, once you have installed it, it will show up in your plugins menu. And so you go to Netscape. And so I think the most useful place to start is, first of all, like I said, it will prompt you to register. And I'm not going to do that. Then, there is this About window, which I'll just show you quickly. It says, what version number, release, and so on and so forth you have. And you can just, dismiss that. And that's where you can get that sort of information. And then, so I'm just going to select the first sort of item here on the menu, which says Build Network. Okay. All right. Oh, good. Somehow it was not taking that long when I was doing it, when I was practicing it. So I got scared for a minute here, but that's all right. So notice that, what happened. We had some tabs here before. And what happened when I opened this, this new panel got opened here, and this is a Netscape specific panel now. Okay, so and I'll just quickly go through some options that we have here. First of all, there is an option to select an organism, human, rat, or mouse, as we sort of discussed earlier today, right? So for now, I'm just going to stick with human, and so this here will bring up this window, and that is where you select which files you want to load into Cytoscape and Netscape. So I'm going to cancel that just for a minute because I want to show you a few things before we load the files, okay? So then, there are these multiple windows here. Let me just do this real quickly. Okay, so this is where you can sort of load compound data, metabolite data, and that's where this data will appear. Genes. And so there are some options down here, which I will go over very quickly. So I mentioned before that you can just add a compound from here. Just type it in. I'm going to add glycine, for example, and show you what happens if I just type glycine. Notice that I don't know anything. I don't know any IDs for this compound. Like, I don't know what the KEG ID is or PubChem ID is or anything else. I'm just typing glycine, okay? And so when I do this, this other window pops up. And so here's glycine, right? And so there is a drop down here of potential matches. So what it is doing at this point, it is simply searching through the list of synonyms that we have in the database. And it is finding anything that contains glycine in the name, okay? But I'm happy with just glycine, okay? Also, let me just show you something. So if I change it to a different compound, so for example, dimethylglycine, notice that the KEG ID changed when I did that. Okay, so let me go back to glycine for a minute. This is a live link, so if you click on it, it will take you to KEG. And we'll open up a page in KEG database for this compound, okay? So anyway, since I was looking for glycine, I can just say okay, and what happened was it added it to the list here, okay? And so it gave me this ID, which it found, which these IDs that start with a C, they happen to be the IDs from KEG database, so this is a KEG ID. And once the compound appeared here, I can actually use it to build the network. Okay? And so I had this slide, which had been shown at several different points yesterday and today. And I talked briefly about different types of network graphs that you can build. And that is this network type drop down over here. Okay? So what this specifies is what kind of nodes will be there in your network. And the most detailed one, as I mentioned, is compound reaction enzyme gene network, and I'm just going to leave it at that. And I am going to build a network using this one compound glycine in just a second. The only thing I wanted to point out is also there is this radio button here. It says use compounds or genes, and what it refers to, use the input compounds, okay? And the alternative, if I select a different radio button, notice that this is grayed. This is grayed out now, and if I select it, then this drop down becomes active. So what I can do is I can select a pathway from a list of pathways now, and so you can select anything you're interested in, such as TCA cycle or whatever. Okay, and so. For now, I'm just, like I said, I'm going to build a network for glycine, and this is very fast because there's just, one compound, okay? So I just did that. And let me explain what we're seeing and what happened now, okay? Several different things happen, okay? So first of all, here's our network, and so I have a mouse, and I'm just sort of rotating my mouse wheel, but you can, without a mouse, you can sort of right-click and, zoom in and out and so on. So it's very intuitive. I think that part is relatively easy to figure out. But anyway, so I can zoom in and out so I can actually, see different types of things. And let me just show you several sort of, you see a variety of different shapes and colors and things here. And let me explain. First of all, glycine, our the compound we searched for is right here in the center, okay? So then let me point out that all the compound metabolite nodes have this hexagons. So they have this sort of pink color, and they're hexagons. Okay, glycine, on the other hand, is red, and the reason for it being red, we wanted to stand out as your seed compound, the query that you started from. And when it's one compound, it's really not that important. Where it becomes important is if you input 50 compounds and you get a large network, then you want to see where your input where your seed nodes are, so it would be easier to identify them visually. That's what we did. Okay. All right. Now it is connected to a bunch of different things, and you can see that all these edges. They have arrows, and some of them have double-headed arrows, and so on and so forth. Okay, so let's focus on this one, for example. Okay, so this gray hexagon is a node that represents a reaction. Okay, and so I will. So by the way, one of the features of Netscape is that you can click on any node, such as this reaction, for example. Well, if you mouse over, it just pops. The label pops up, so it is easier to see, especially if you're zoomed out a little bit more and cannot read it. It's a useful feature. But if I double-click it right now, this information panel pops up here, and it has information about this particular reaction, and I can sort of move it so you can easily you can see it more easily. What this particular. So here is an equation for this particular reaction, so you can get more information. So this is not, according to the database that we have, it's not a reversible reaction, and there is this additional information about reactions. So I'm just going to close that panel for now and continue sort of reviewing these different shapes. Okay, so now this, and I can sort of move it around, or so it would be easier for me to see this. So this is a reaction. Here's another compound that is connected to this particular reaction, and this happened to be sarcosine. And so the arrow indicates that the product-substrate relationship, and so sort of flux through this reaction, if you will. Okay, and here now I'm going to zoom in a little bit more. Here is yet another shape and color. It's another rectangular shape with these rounded corners. So this is an enzyme now, okay, glycine and metal transferase, and it is connected to the reaction node, and another node connected to it is right here, and this is a gene that is encoding this enzyme. And in this particular case, it appears like there is a one-to-one relationship. Yes, please. So the circles are genes. I'm just sort of clicking on it, and it shows here that this is a gene, this is entree gene ID, this is gene symbol, gene description, subcellular locations, and so on and so forth. So far we have 4 different types of nodes. Now, what I will show you a little bit later when I load gene data, just like, normally if this, okay, awesome. Let me back up a second, okay? So remember my input compound was glycine. So all this sort of, relatively big network was built just, from one input compound, right? So where did all the data came from? Obviously, it came from the database, right? Because as I said before, we store all these relationships in a database, okay? Now, the only compound that came from our input was only information that we gave it was glycine. So that's why there's bright red. Now, if I were to give it the input set of genes, those would be shown in navy blue as opposed to this pale blue color. And that's how we decided to sort of, visually represent those. And so any sort of, ideas on sort of, better visual representation are always welcome. And so any thoughts will be, are sort of appreciated. Okay, so any questions so far? Okay. So, right. So the data that we're displaying here comes from the database that we used to sort of, draw these relationships. The Edinburgh Human Metabolic Networks, they have the subcellular locations, and the subcellular locations are associated with reactions. That's part of the data that we got from that database. Okay? All right. That is exactly right, and actually that is where I'm going next. Okay, so let's explore this new sort of window or new tab that popped up when we created this network. This pathway filter wasn't there before, right? And what it is doing right now, let me just close this because we don't need that, and let's look at the whole network. Okay, so these are all the reactions and all the compounds involved in these reactions that in our database, that exist in our database for glycine, okay? And so these now here are the pathways in which, so these are the pathways for those reactions that glycine is involved in. So we call it a pathway filter, and it works as a filter. So you notice that I just clicked on bile acid biosynthesis. I don't know if you can read that, but I unfortunately cannot zoom on that part. But anyway, so what happens if I click and select one? Everything that belongs to this pathway gets selected on the network up there. Okay, and so these are all the pathways in which glycine is involved. So basically, I want to point out one very sort of important philosophical sort of, thing here, okay? So it's not pathway-based. It's really network-based. So our idea was, we didn't want to do, like, we didn't center it like on one pathway at the time. We wanted to build a network, okay? Which can go across different pathways. I mean, we can build a pathway, sort of network, pathway-specific network too. I showed you a drop-down where you can select a pathway. But this is driven by your input data, okay? So if, in other words, if the tool can connect different compounds that you give it into one network, it will. It will try that, at least, okay? If not, it will show them as disjoint networks, yeah. Right, so what I picked here are things that belong to urea cycle, and the easiest way to explore that actually is to click this button to create subnetwork, and that's how you very quickly can subset very large networks. That's what Chuck described and I tried to describe in my talk earlier, okay? So this particular subset of compounds and reaction belong to urea cycle. Now, is this the entire urea cycle? No, of course not, obviously, right? And so what Chuck said before, and I don't know if, well, here's a good example, right? So one of the functions, very important functions of the program that I want to show you, okay, notice what I'm doing. I actually right-clicked on the node, and I go down to Netscape menu, and I have an option to expand this network, expand an existing network, or I can expand in a subnetwork, will pop up another window, but I'm just going to expand an existing subnetwork, okay? And what happened here, it just, expanded this network from glutathione, and so the new nodes there were new parts of network which were not there before. Okay, so the edges are blue, and the nodes are this sort of, I don't know if you can tell the difference, but they're kind of this more like pale color. Okay, and so what you can do is you can just sort of keep expanding that network in the direction that you're interested in to bring more compounds into the picture that you want to look at. Okay, so so far we have not we have not put in any experimental data. We just played with the software and built some networks, but we haven't done anything with the experimental data, and that's what I want to show you next. Okay, so I think what I'm going to do, I'm going to show you. Okay, so there are all these different tabs here, and I think it's important to know. Unfortunately, they don't fit. I can make this bigger, but I don't want to, so I can just sort of move it a little bit, right? So this tab here, which is called Network, has a list of the networks, and this is a subnetwork that we just created. So I can just go here and I can say, okay, well, I'm not going to look at that network again. So I'm just going to say destroy network, and I can just eliminate them, and then I can go back to Netscape panel, and I'm going to just say clear. So I just want clean slate. I don't want anything in here, right? And I'm going to close the pathway filter. So, okay. So now, so basically, hopefully we have clean slate, and I can select experimental files. Before I do that, actually, I would like to go to this slide for a second and briefly tell you about the data that I'm going to load for the demo. Okay. And this is the published sort of literature-based data set that we used in when we published Netscape tool. We used that to demonstrate the functionality of the tool, so I like to use it. So gene expression data and metabolomics data both come from they come from two independent publications, and so these are from pancreatic cancer. So gene expression data set was quite large. There were 36. Patients' tumor and normal tissue samples from each patient, and so the data were downloaded from geodatabase from public domain, and this is the publication. And the metabolite data set is much smaller, and I am going to just rather than showing slides, I will show you. I have the same data set open in Excel. So the metabolite data set is quite small. There are only 12 compounds. All 12 looked relatively significant. I only took the most significant compounds. And so this is the basic data format. Notice that in this case, I rather than using names, I'm using what are called KEG IDs, because I don't want to go through this drop down and sort of disambiguate every synonym and so on and so forth. So I just took KEG IDs. So the second column are p-values. Also, I mean, basically it can be any significance value. It doesn't have to be a p-value. And this is fold change. And what I'm showing here are, so obviously, these were up and this went down. So I think it's sort of pretty self-explanatory. Now, I'm going to show you the gene data. And so this file is actually quite large. So this is like, from my microarray. So, these are all the, so I think they're like, what, 40-some thousand different genes. So it's quite large. So it's going to take a little while to load that file. And I'm just sort of warning you, okay? So these are, so the identifier for the genes here is untrade gene ID. And so this is a p-value, and this is actually q-value adjusted basically adjusted for multiple comparison. And what I want to point out that, a lot of times what is used for both gene expression actually in metabolomics data rather than looking at fold change, a lot of times people look, well, especially for gene expression, log fold change, and the program should be able to handle this sort of thing. Okay, it does not matter how many columns you have in a file, because when you load the data, you will be able to map your column, and I'll show you how to do that. Okay, and one more thing that we did with this gene expression data, with this file, I mentioned this program LRPath, and so what we did, we actually ran this program to find enriched pathways. Okay. And that's what the output of that program looks like. So this first column here will be a pathway. And then, okay, well, let me explain something. I've used pathways and I have used concepts, and I just want to kind of explain this a little bit to avoid confusion, okay? So when you do enrichment testing, you can test against any sort of, predefined biological categories. This can be pathways. This can be, I don't know, any other groups. So I can define any arbitrary groups of metabolites or genes, and I can use them in enrichment testing, okay? And so we're kind of, avoiding the term pathways here, or, so we prefer to use term concepts, okay? What is also commonly used, if you analyze gene expression data, then you will know that gene ontology terms or gene ontology categories are often used in enrichment testing. And these are concepts. I mean, these are entirely valid. It can be anything. So we made it sort of, we wanted to be able to put any concepts, not just pathways. And that's why they're called concepts. Okay? So, anyway, so this p-value here now in this false discovery rate, this now refers to the significance of a particular pathway. And that file we got from gene expression data. And that's what I want to load into the program. Okay? And so if I go back here now, and then, okay, here, this drop down essentially, duplicates for the organism. So this data are from human. Again, I'm not going to change it, but if you forget to change that and try, loading the data from wrong organism, it will give you an error because it will not be able to map those genes.
So where do you generate the analysis for your pathway, for your Excel, the last Excel that you were talking about? So where do you go to generate?
Where did I go to generate that? Okay. Well, I'll get to that in a minute. Okay. I'll show you. Okay. So let me just, go here first and select this metabolite file. Okay. Metabolites. Okay. So notice I told you that you will be able to map the columns in your file to these fields, and so that's where you select. So p-value, I'm going to map to p-value conveniently, and fold change to fold change. And when I do this threshold here appears, and you can set the threshold of what you call significant. Okay, so the threshold, like p-value of 0.05 sounds okay, and I'm just going to keep it at that. So anything anything that has p-value below that is going to appear is going to be visually sort of represented as significant. Okay, that's where I set it. Okay, with a fold change, I can also set some significance threshold, but I'm not gonna. I'm just going to keep it at 1, and so fold change of 1 means that it have not changed. So I'm not going to worry about that just yet. Okay. So now let's go to and import the genes file that I just showed you. And I think it's going to take a minute because it's a very large file, as you saw. And while it is doing it, I'll just sort of explain something. Okay, so you can put a whole sort of large gene file like that, but if you're not going to put the concept file or file with enriched pathways, I wouldn't recommend doing it. Okay. And the reason I don't recommend it is because if you're going to try and build the network with like, whatever, 40,000 genes or whatever you have, if you have a lot of memory on your machine, Cytoscape is powerful. I mean, you may be able to do that, but A, you won't be able to do much with it. And, A, it will take a long time, and B, you won't be able to do much with it. So I don't recommend it, okay? So if you are not planning on putting any enriched pathways in, you may want to just, limit it to the most significant genes or, some smaller subset of genes that you have defined. So just be a little careful there. Okay. Now, the question was, how can you generate that sort of file of enriched pathways? Okay. So there are two options for doing this. I can import that file that I just showed you right here, which is what I'm going to do today. Or what I can do instead of doing this, I should be able to run that file right. Run that program LRPath right from here, okay? Which I'm not going to do because it's going to take a long time. What I will show you, though, is this. We have a different website, and we also published it, okay? So it also has this sort of web interface now. So the address for that is lrpath.ncibi.org. Okay, and it gives a little overview and more information about input format, so you can run it from here separately and so on. Okay, so it's just a little sort of, we got a little sidetracked. For now, I'm not going to run it. Like I said, I'm just going to take that file that I just showed you and put it in here. Okay, so now I'm loading a lot of data, so this is going to take a little while. So it would be a good place for me to kind of, while it's doing it, I can stop and, answer any questions that you might have or whatever. And so the rest of it, I'm actually going to do a little bit like, like they do the cooking shows on TV. So, you do something here, and then, you go to, like, voila, here it is, like, already prepared. And the only reason I'm doing it, is because we would have to wait a little while. I'm loading a lot of data, okay? So, but this part should be relatively quick. What will take a long time? So right now we will just sort of, load the data, and hopefully it will populate these two fields for compounds and genes over there. Oh, okay, it just got done. But there is another step that takes a while, that is actually building a network. You remember for glycine, it was very fast, but this is going to be a little longer. Anyhow, okay, so one very important question is when you are sort of doing any sort of mapping, and I told you that, not everything can be mapped. Obviously, there are some genes or some compounds that were not found, right? And so Netscape is very diligent about reporting this back to you. And so what you can do with this, it gives you an option to save that to a file. And I actually recommend doing this, okay? So at one point in time, we had it so you can, call that window back. But for a variety of reasons, we decided that it was not a good idea. So this is your chance to save that data. If you say OK, it will just dismiss this window and you won't get it back. But for now, I'm not. It just will save it into a text file, okay? For now, I'm not going to worry about that. But just to show you some genes, quite a few genes. But if you consider that we were putting in like some 40,000, maybe it's not that bad. But and 4 compounds were not found. So I'm just going to say OK. And so all the genes and compounds that it was able to map, it put them right here. By the way, you can undock this panel and just make it just a little bigger so it is a little bit easier to see. Okay, and so or to put it back, I just clicked on that corner and it's back here, right? So now I'm just going to click build network. Actually, I'm going to pretend that I'm doing it, and I'm going to go to another instance of Cytoscape where I did that before. And that's where the cooking show part comes in. Okay, so what would happen was this, okay? So if we would, wait for maybe, and it's not that long, it's just, a little too long to do in real time, okay? So it has this very, very large and, very sort of dense network. And, you can't really make much sense out of it. But, if you zoom in close enough, you will notice sort of, the same shapes and the same colors that I was talking about. So, the one we're looking at here, and notice, by the way, that when I select a compound, it becomes yellow. But so this is bright red, as I told you before. It means that it was in our input data. And so this is cholesterol. But notice something else. So that something else is this bright green border around it. What it means is that it was significant. Okay? It passed our the threshold that we set. Okay? If it wouldn't be significant, then it would not have that green border. Okay? Now, also, you may notice that this node here is a little bit bigger than, other pink nodes around it, okay? And the reason for that is that the fold change was positive, okay? So the compound went up. That's why this node is bigger. Same is true for this gene. Like I said, the genes that were part of our input data set are blue. And so this particular one went up, but it doesn't have a green border. Therefore, it did not pass our significance threshold. This, on the other hand, is smaller, and it went down, but according to our threshold that we set, it was significant. Now, there's something else very important that I want to show you, and that is, so notice when I'm zooming in and out, I want you to look in this corner and see what is happening, right? So do you see this? This is this rectangle, okay? That's where you can sort of, navigate your network and select. Notice that my mouse is over here in the lower left corner. And so as I'm zooming in, this basically, shows me the area of network that I'm looking at, okay? So there are quite a few genes in our input data set, okay? So let me stop here, actually, because I don't know if I made it clear. So you may notice that I don't know if you noticed that before or not, but you may sort of, see that. So, okay, so this gene, this node is like pale blue, but yet it appears to be significant. And so, if we have data for it and it was in our input, how come it's not navy blue, okay? So the reason is, and I kind of alluded to it earlier, but I don't think I made it clear, okay? So when you are loading gene file and concept file, okay, the program only takes into consideration, so it treats as input genes that were in the concept file, okay? And so if, in the process of building network, it pulls other genes from the network, and there are experimental data for them, it will take that into account, if it makes any sense, okay? I think it will take some getting used to. So the only thing I can tell you right now that it isn't described in some detail in our manual, okay? And so what I want to go to is this panel down here, okay? So what do we, yeah, sure.
Understand how if it was not in our data set, how is it significant?
Okay, sure. Okay, it was in the data set. It was in a big gene file which had 40,000 genes. But it was not part of any enriched pathway, okay? It was not one of the genes that were in these enriched pathways, okay? So what program is doing, it is pulling it starts with a set of genes that were in that concept file, okay? And then it is going to a database, and it is pulling the information about other genes, compounds that are connected to this experimental compounds, okay? And at that point, if we happen to have experimental data for any of these pooled compounds that were pulled out of the database, it will then, display that experimental data for them. So it is doing several rounds of sort of, checking and validation. I'll show you where experimental data comes from in a minute, okay, when we get to that. I mean, we have worked on it for a long time. It does have a lot of features. I mean, I think you can navigate it successfully even if you kind of, don't look at genes, for example. But I thought it would be useful to show it. Maybe I went too far with that, but I promise I'll try to make it clear. So anyway, what I wanted to review here is, so this is a pathway filter, much like what we saw with glycine network. And it just, lists all the pathways that, found in this data up here, okay? And the same rule, the same thing is true. So you can click on, one or more pathways here, and, respective nodes up here will get selected, and you still can create subnetworks, okay? It also popped in this additional tab here, which is now called, okay, this was pathway filter, and this is now called concept filter, okay? And the content of that is essentially the same that was in this input file for concepts that I showed you, okay? And one of the things that you can do with it is, if you go back here, you can sort by any of this column, like, false discovery rate. So I can just, sort it such that the most significant concepts are up here. And this seems to be, all of this seem to be, considering that this is an adjusted p-value, this seem to be pretty significant. So, for example, I have selected glycine, serine, and threonine metabolism here, and I actually, beforehand, I actually created a subnetwork, which is right here. So I'm just going to that subnetwork here. And so now I can just, look at these rather than looking at the whole dense network, if that's something, I'm interested in, or if I suspect that it may be related to, something that, the pathway that may be of interest and for the systems that I'm studying, and so on and so forth. So, one of the things that I see here is arachidonic acid that went up and, apparently was significant in the number of enzymes. Phospholipases here, they all, like, there's a whole bunch of them, and it seems like they all went down in this particular data set. Okay, so that's the sort of thing that I can quickly do with the program. And then, what I can do, like I said before, I can double click on any of them and I can get more information. And this will now take me to KEGG database or to Entree, where I can get more information about a particular gene. And so that is all here. Right, now about experimental data and about also something about Cytoscape that I need to tell you, because this is a Cytoscape feature. So far we looked a little bit at the concept filter and the pathway filter, and this is something that is Netscape specific, that we built essentially. Okay, these first three tabs here, these are the Cytoscape features, okay? So let's concentrate for a minute on this, which is called Node Attribute Browser, okay? So right now, when I clicked on it, basically, what it has a list of all the nodes in that network that I have selected here, right? But there is this little icon in the corner, and it says Select Attributes when I mouse over, and I'm going to click on it. And what happens is now it's going to list all the available attributes. And so among those, there's a whole bunch of different attributes for compounds. So, for example, if I wanted to look and see the compound names here, okay, so among other things, I may want to see the experimental data for compounds. So what I'm going to select now, compound metabolite, metabolites fold change and metabolite p-values, okay? And then, when I click here, what happens is that it actually lists all these columns that I selected here. So I can, by clicking on a header, I can just sort them. And so this is a compound, so I sorted them alphabetically, in alphabetical order by compound name, but also I can sort it by compound p-value. And what this will do, this will just, show me in this particular subnetwork, there are 3 compounds, And these are their full changes and p-values, and these are the three compounds, phenylalanine, tryptamine, and arachidonic acid, okay? And so all kinds of information that you see, I mean, I don't know why I wouldn't necessarily want to, but if I want to, I can just, copy and paste that into an Excel or any kind of file or any other sort of, application that I want. Okay, that's how you can quickly get to the experimental data. And if you wanted to see what these values actually are. Because I guess, one thing that I want to point out is that we don't visually represent in these networks the amount of the change. We tried that. And so you can sort of quickly tell that, arachidonic acid is up. But from this, you cannot tell whether it's, 5-fold change or 2-fold change or anything like that. So again, we tried different sort of options there, but it's extremely difficult to map fold change. I mean, basically, as it turned out, visually it's very difficult to tell anyway how much it changed by just by the node size. And at some point we ended up with huge blobs and something so small that you cannot see it. And so even in a log scale, so it just didn't make much sense. So right now, the size of the node represents just the direction of the change, not the amount of the change, okay? So let's see what else I can sort of, show you here. Okay, well, any further questions? Yeah.
When you put in one compound, is it only showing one reaction away from the compound?
Yes, it goes one step away. That's correct. That's correct. And you can expand that manually to see, like, just keep walking the, particular direction. Yeah, you can keep expanding. And by the way, if you sort of, expand it and you want to go back, you can collapse that network with expansion nodes. And I think it's sort of, pretty good at keeping track of sort of, these expansions and collapsing them back. Okay, well, another important thing is that you can save. So if you have done all this work and created these networks, and then, you have to, close it and just, to come back to it tomorrow, what you can do is you can save your session. And the way to do that is you go to a file and you can say save or save as, and it will save it into a site escape session file. And the extension of that file will be CYS, and to go back to it when you have a saved session. Okay, so just remember when we sort of started doing this, we went here and we said select files, load experimental data, and so on and so forth. Okay, so if I wanted to start a saved session, I would say I would go to File, Open. Okay. And it will warn you, okay? So it can't have two sessions opened at the same time. So your current session will be lost. Do you want to continue? In this case, I don't, but that's how you would open a new session. And again, we put a lot of effort into it. I think it's pretty good most of the time about saving the networks that you have created. But I think, sort of, my advice is if you came to a point where you think that this network is useful, I think it's better to basically, save the image or whatever it is that you might need for your presentation or publication or something like that. And something about saving, okay, let's see here. Site Escape itself has a rather detailed manual, and there are some useful instructions on how to save images. Okay, well, to be quite honest with you, I guess, I mean, it's not the easiest thing to do to, okay, well, it's a little bit of a challenge showing these networks. Okay, most journals require, if you plan to submit it to sort of publish it, it has to be certain resolution, and just if you just do a screen capture for a PowerPoint presentation, it will do very well most of the time. But it will not be sufficient. Chances are that it will not be sufficient for a journal publication, okay? There's a way to do that in Cytoscape. It is not particularly elegant, but there is a way to save a higher resolution image, okay? So, I mean, if you have any questions, I'd be able to, like I said, it's not particularly elegant, but I'd be happy to advise you on how to do that if it comes to this. So hopefully, you know, if you find the tool useful, then, you know, maybe you will want to save some data someday. I'll be happy. But I mean, also what doesn't work very well, I mean, I think a lot of times what I've done, we had to do was to, I don't know, put some labels in, you know, afterwards in some sort of, you know, PowerPoint or any other sort of, you know, program like that, and sort of sometimes relabel the notes, because it's what is very easy to see here is really not that easy to do, you know, when you have, I mean, here it's like, you know, even if you go to a big network like this, you zoom in and out, and sort of it's almost like instantly you can see the details, and you know, if you try to create a sort of, you know, figure, it is not always that easy. Okay, so any questions?
I missed the part because I had to go out. I missed the part of importing the data from for example, from a Excel sheet. And if we have different treatment groups, so what will be the, shall we generate 4 different maps, or how you organize it?
Well, the program is rather versatile in that respect. So you can do a number of different things. Okay, so what I showed you, okay, so so there are several assumptions that have been made here. Okay, so one assumption is that you have already done statistical analysis of your data. Okay, and so if you have different groups, so for example, let's just take the simplest. Let's start with a simple case when you have 2 groups, control versus treatment or something like that, right? And we're talking about specifically about metabolite data. Let's not worry about gene expression or anything like that. Okay. So if you have these 2 groups and you have already compared them, then you may already have fold change or log fold change or something like that, and you already have some kind of significance, you know, value. You can have, you know, p-value or something like that in the examples that I was showing. Okay, that is the simplest case where you can just, you know, put that, and that would be your input file, just like I showed here. Let me go to this Excel file again and show it to you. Okay, so I have compound ID, I have p-value, and I have fold change. Okay, that's one case. Okay, now Chuck showed you this example with animation where clearly there were, like, you know, different treatment groups and so on and so forth. And in that case, the workflow would be slightly different. Okay, so you can have this multiple sort of, you know, measurements. In that case, you wouldn't have fold change. In this case, you would have the actual, maybe normalized measurements for, let's say you have 3 different groups, you know, control and 2 treatments, and each of these has 3 or 5 time points, this type of data. Okay, so you can put all this data in, and I wasn't actually planning on showing that, but that's how you would get to this animation. And there is a way to do that here. There is a separate menu under Netscape menu right here. Okay, it says, where it says animate, what would happen is it would pop up this panel. And so basically it would allow you to, I mean, I don't have the right file loaded for that, but if you had these multiple columns, that's where you can sort of tell it to build, like, you know, if I had, like I said, let's say we have 3 time points and 3 control and 2 treatments, right? So that's where you would tell it to build basically a 3x3 table, and you would tell it, like, you know, which sort of, you would designate your rows and columns right here, right? And so then you can just sort of, you know, drag the names of the columns here, and that's how you would build your animation. I think I might have a file like that on my computer. So I think we can. I'm happy to stay afterwards and show you if you like.
Is from ANOVA, or is it p-value of correlation? From where?
Okay, well, for this, in order to do this, you do not need actually p-values. You actually need your experimental values, right? That's why I cannot show it to you with a file that I have. I mean, you can have p-values from ANOVA, but okay, there are two modes in which you can use the program, right? So you can have, if you're looking at p-values and fold changes, you can only have two groups. But if you want to build this animation, then you can have as many groups as you like, but then you have to have individual measurements. If it makes any sense. I'll show you the data example, and I think it will be easier to understand then. I'll look, but I'm pretty sure I can, or we can create a fake file or something like that. I'm pretty sure I have on my computer. Any other questions? So this is a lot of information in a very short period of time. I think it's not as complicated as it sounds. And like I said, there is a rather detailed user manual, and there is a video tutorial which pretty much goes step by step through what I described to you. And we salvaged the link to that, which mysteriously disappeared. What? It is already working. We already put it back. The video tutorial, I don't know why link got lost. Yeah. It's online. Let me go and show you real quickly here. Okay, so you're on the Netscape page. So if you, this is the home page that you will get, and this is this help tab. Okay, so that's where it is. So I think I will. So basically, you can just play a movie here. And I have the sound off, so, but there will be a sort of, movie, and it will show step by step how to do that. Okay, well, if you have any questions or, want to see any details, I'll be happy to show it to you and, talk to you and figure out what other things we might want to do with it. I mean, I guess, the only other thing that I might show you with this data set, if I still have time. Misty, do I have time? Not really, okay. Okay, well, let me, okay. Okay, so if I go back to Netscape panel, I just sort of maybe, it won't be very quick, but we can try. Okay, so I still have all this experimental data loaded, so I wanted to try and see what happens if I try to select a pathway and show you a pathway now. So this is relatively small network. So this is now like a whole TCA cycle loaded here, but the thing that I wanted to point out is that it knows about the experimental data we loaded. So the genes that, in this case, the genes that were, up or down in your experimental data that you loaded, they will still show up in that pathway display here, if it makes any sense.
So here it's still selecting only the compounds that were in the experimental data?
Nope. That's what it did differently, and that's why I wanted to show it. What happened was, instead of telling it to use the input genes or compounds, it is now using the set of genes and compounds in a pathway that I selected. But still, okay, it still keeps in the memory the data set that we loaded. It's just, selecting what will be displayed in a given network. And that is determined by this pathway drop-down now. So I guess, okay, here's what I want to leave you with. So it's all about, like, you load a lot of data in, you pull a lot of information from the database. And from there on, it's about slicing and dicing and filtering, okay? So in a nutshell, all we have is a bunch of different filters, okay? And a bunch of different ways to kind of slice your data and, look at, different parts of the data and, different pieces of information that we pull, okay?